Greetings, welcome to Brain Gauge channel. I'm Mark Tomerdahl, and in this video, we're going to talk about the brain gauge metric accuracy. And the best way to think about accuracy is your brain's ability to contrast enhance inputs. In other words, if you've got a contrast knob on your old television, you tune it, or if you've got a contrast adjustment on any monitor, you tune that into some specific point. And you're basically, your brain is fine tuning on an input. So that's one way to think about it. But where do you see that in the brain gauge? In the analysis app, it's right here. This is the accuracy score, and just a reminder, this is someone's overall trend timeline right here. And on this data point here, we've got all their scores. Accuracy comes from is calculated from amplitude discrimination or sequential amplitude discrimination and simultaneous amplitude discrimination. And in this case, these people had DLs of 25 and 32, which are very good. DL is difference Lyman, and I'll explain what that is in a minute, but a little bit more. But anyway, these are the two values that are used to generate that number. And something to think about is that if you ever do want to go look at these raw scores, they're right here on the right-hand side of your analysis app, and you just scroll down and look at them. And basically, smaller numbers are better. Over here, bigger numbers are obviously better, but they're scaled to 100, and that 100 is in this normative range. So anything from like 20 to 50 is about in pretty solidly in that normative range. 20 is a 10% DL, and you can't really do a whole lot better than that. That's really pretty much as good as it gets. You might see one or two super testers get lucky and get a little bit below that. That's actually a very good score. All right, let's see what else we got. So for the short answer, people who just want to read this and then get it heck out of here, there are populations that do very poorly on the accuracy metric, and they're listed right here. And basically anxiety and depression and neurodevelopmental disorders, neurotraumatic disorders such as head trauma, chronic pain, neurodegenerative disorders such as Alzheimer's and Parkinson's. All those groups do poorly. Now, if somebody does poorly on the accuracy metric, it does not mean they have a neurodevelopmental disorder. If someone has that disorder, they probably will do very poorly on that metric. That's the way you think about it. It's not a diagnostic, okay? So we don't want to get in trouble for saying it's a diagnostic. All right, so let's get on to exactly how that value is obtained. Well, what we do is we deliver two stimuli, one to one finger and one to the other. And notice these are shaped, these little sinusoids. That is the shape of the stimulus that's delivered to those fingers. And basically, you can see that this is larger than this stimulus. And the subject is simply queried, which one is bigger. That's as tough as the questions get. And at the very beginning, the difference between these two is very big. Just like reading an eye chart, extremely easy to tell the difference. And, what happens is, like in this case, in this particular graph, the biggest one had a value of 800, smallest one had a value of 400, and that was very easy. So somebody gets it right, and then they go to the next, and each time they get it right, it gets harder. So that difference gets smaller and smaller and smaller. Now, the final difference that you see over here, that final difference between this number and that number is the final difference Lyman. Now, in the brain gauge that you have, unless you're doing some kind of research, these numbers are typically 200. 200 versus 400 is a starting point, and roughly, like if I mentioned the last person had a DL of 20 or 25, that's about a 10% difference. So the value that the 200 was being compared to was 225. That was their final value was 200 versus 225, and their DL was 25. So anyway, in this case, this was 456 versus 400, or 56 over 400. That's a little bit over 10%, 56 to 400. So anyway, very straightforward, and people generally track down fairly well. Just to clarify the difference between simultaneous and sequential amplitude discrimination, what we have is called a test stimulus, is a larger stimulus, and a standard stimulus. Now, it's random. The larger, which one is larger, is obviously randomly delivered to digit 2 versus digit 3. So it's not always the same answer. Trust me, there were some pretty old protocols 50, 60 years ago where people sort of did that kind of thing, but that's okay. We won't go into that. So anyway, test stimulus is large, standard stimulus is small. That's the correct answer. That's simultaneous, same time. Sequential, exact same protocol, except one stimulus happens long before the other stimulus, half a second. And so they're not being delivered at the same time. And basically that just means there's not as much competition between the two adjacent stimuli. If you're doing a very short battery, if you're aiming for a very short battery and you just want to do a couple of tests, you just do the reaction test and say one of the accuracy tests, you can get an accuracy score, but I would suggest sticking with simultaneous only. That's a much harder test to perform well on in general, and it's much more dependent on lateral inhibition. Anyway, that's my suggestion. Now, what's the basic hypothesis that goes on with, behind this? It has to do with lateral inhibition. And the original version of this came from George von Bexey in the 1960s. And he extrapolated this model of lateral inhibition, which is where you excite one area and it's surrounded by an inhibition. Anywhere there's excitation, they'll be surrounded by inhibition. And it's kind of interesting that he predicted this, but based on observations of sensory perception, he did not stick any electrodes in brains. So he really didn't have firsthand knowledge of this. He just predicted it from measures of sensory perception. The reason I emphasize that is because that's what the brain gauge is. It's a measure of sensory perception. And, so sensory perceptual measures kind of led the way, in that took a while before you started seeing images like this. This actually came from our lab. And basically what you see here is area of excitation, this peak, surrounded by this area of inhibition. Why is that important? We'll get to that in a minute. But what happens next is that if you put two stimuli together, you start saying, well, we can do experiments where we deliver two stimuli, and this is patterns of activation in the brain, and you see these two stimuli activate these different areas. And what we did was we actually compared the activity patterns that we got by delivering different amplitudes of stimulation side by side, and this was in an animal. This is non-human. But it was very high resolution. This is a micron resolution. And then we compared that with measures of sensory perception. And those comparisons correlated very nicely. Okay. So anyway, we've got, straight lines are good when you're looking at correlations, by the way. Anyway, what did I mean by contrast enhancement? Contrast enhancing the brain. So you think about it, if you give just one brief stimulus, one brief sinusoid to the skin, you get this percept of very broad-based stuff. And actually, von Bexey did some of this too. And then when you deliver multiple stimuli, that pattern starts shaping up and gets very sharp. And that's why we have a specific stimulus duration that we deliver with the brain gauge. We deliver specific stimuli that we know will make this type of pattern, and when it makes that kind of pattern, it plays a big role in the contrast enhancement. For example, if you look at activation patterns in the brain and you see these two areas right here, and you activate this area and you activate this area, you get very clean contrast, very well contrasted between these two areas. Now, if we turn off the lateral inhibition, just turn it off just a little bit, and this is done with a GABA and GABA antagonist, where you actually are turning off that key feature of the lateral inhibition. You basically see a picture that's getting fuzzy. You're losing contrast. And this is actually what happens in a lot of those neurodevelopmental disorders that we listed a little while ago. And so let's list them again. So now you got the reason why these populations don't do well on this particular test. These are populations with low levels of GABA. People with anxiety, anti-anxiolytics are usually GABA agonists. Depression, panic attacks, stress, insomnia, those are all, they all have some effect on overall GABA levels or GABA efficacy. Neurodevelopmental disorders, autism, well known for having low GABA. ADHD, Tourette's, OCD, epilepsy, head trauma, chronic pain, and neurodegenerative disorders. A lot of people are very interested in aging and aging dementias. All that traces back to lower levels of GABA. You say, well, how can one neurotransmitter have all this to do with it? Actually, this is a predominant neurotransmitter in the brain for inhibition. So there's GABA A and GABA B, and there's actually GABA C, but you don't find that. No, I don't think anybody's found it in the brain yet. It's in the retina. But anyway, everything asterisk, there are publications using the brain gauge methods. Anyway, that's just a very short primer on the accuracy metric, and hope you got something out of it. If you didn't, write me a note and I'll try to redo it and make it a little better. So that's all for now. See you next time.